Hello everybody, it's Mrs. Pound and we are studying Chapter 7 on Biotechnology from BJU Biology's 4th edition. The section where we're currently working on is Section 7A on Biotechnology. And today I want to talk about subsection 7.2 on genetic engineering. So our objectives today are to define genetic engineering, describe the steps to produce recombinant DNA, and describe how recombinant DNA is used in bacteria to produce human insulin. So genetic engineering is the manipulation of chromosomes or genes by artificial means. This can involve a single gene being taken from one genome and placed in another genome. So from one organism's genetic material to another organism's complete genetic material. This modified DNA is called recombinant DNA. Notice recombinant looks a lot like the word recombined, and that's what it is. It's recombined DNA. So recombinant DNA is DNA that has been formed by placing two pieces of DNA from different sources together. This is accomplished using restriction enzymes. These enzymes snip DNA at certain base sequence. So they're like chemical scissors. And just like this string, it will find a base sequence, though, that it recognizes and snip it at that base sequence. Now, these enzymes, these are not something we have created. They are found in nature. We just discovered them. God created them, and we make use of them. And when they cut the DNA, they actually leave sticky ends that will stick to DNA that's cut with those same restriction enzymes somewhere else. So the restriction enzymes are used to snip genes out of chromosomes and insert them into other chromosomes using what is called a vector. And a vector is the agent that is used to carry a new gene from one organism to another during genetic engineering. One common vector is a plasmid. This is a small circular piece of bacterial DNA. So here are plasmids. And plasmids in bacteria tend to be used for reproduction. These plasmids already do this. Bacteria can exchange genetic information using their plasmids. So a plasmid is important in genetic engineering because of its role as a vector. So we take that DNA fragment that we snipped using the restriction enzymes, we cut the vector with those restriction enzymes, we put them together, and it becomes recombinant DNA. We then place this plasmid back into bacteria, and it can be used to manufacture important hormones and things for us. It's things like insulin. So an important body chemical that has been mass produced with the help of genetic engineering is insulin. And we use this process on E. coli bacteria. We insert the gene for human insulin into a plasmid of E. coli bacteria, and the E. coli bacteria produces the insulin for us. So insulin producing bacteria is an example of a transgenic organism. And a transgenic organism contains genes from more than one species, in this case, human DNA and bacterial DNA. Now, this is an important thing that we can do because before we could make human insulin using bacteria, people who were diabetic and dependent upon insulin got their insulin from the pancreases of pigs or cows. And some people were allergic to this insulin, and it was very expensive because they had to take it from those things that were dead, because you're not just going to remove a pancreas from a live animal. So it had to be dead. So it was very expensive, it was tedious, and this is a much better source. So our objectives today were to define genetic engineering, describe the steps to produce recombinant DNA, and describe how recombinant DNA is used in bacteria to produce human insulin. So don't forget your 5 questions in your notes and keep watching the video to answer the questions at the end.